Good morning, everyone. We will be discussing the effects of using CRISPR-Cas9 for gene editing in relation to neurology.
Gene editing is the ability of manipulating an organism's DNA in certain areas of the genome. Gene editing is also known as genome editing. A popular method used is the CRISPR-Cas9, which is short for clustered regularly interspaced short palindromic repeats and CRISPR-associated protein 9. CRISPR is a tool used to cut out particular parts of the DNA once the correct sequence is found. These procedures are done in order to cure and prevent certain illnesses, though there may be some risks or side effects depending on which part is being edited. The goal of this research is to discover the benefits of editing for neurological illnesses or disorders and to determine the risks of doing so.
Editing the gene involving the brain may be risky, though it can be a solution to neurological disorders and illnesses.
The source of neurological diseases involves genetic mutations and abnormalities within the nervous system. Recent studies have shown that brain and nervous system cancers have increased by 8.9%. Brain tumors and nervous system cancers are mainly caused by repair mistakes in the DNA sequence of a gene. In view of that, one of the features of the CRISPR-Cas9 system is that it could locate, edit, and cut out a specific part of a faulty DNA sequence. This method in particular has been used in several trials in order to test its effectiveness. In fact, in the year 2018, scientists have identified antibodies which target the Cas9 protein utilizing the CRISPR-Cas9 tool. In addition, a conducted trial on amyotrophic lateral sclerosis or ALS patients have also shown positive results on cell dose escalation, designed based on the studies presented. Although, as mentioned beforehand, gene editing, which involves the brain, may possibly be risky to certain neurological disorders and illnesses. This is because insertion and deletion of some parts of the faulty DNA sequence can potentially be unforeseeable.
As mentioned in the evidence, recent studies have shown that nervous system cancers have increased by 8.9%. It is imperative to the research potential solutions to these neurological diseases and illnesses. Researching on effects of using CRISPR-Cas9 for gene editing in relation to neurology is significant because it opens opportunities to test whether different types of gene editing would indeed show benefits to curing neurological diseases, or if it would do more harm than good on the brain. With the proven capabilities of CRISPR in gene editing, it is clear that this type of genetic therapy could help improve the effectiveness of other treatments, as well as eliminating patient complications after certain procedures or clinical trials.
The post-positivist paradigm will be used in order to obtain data to determine the benefits and risks of using the CRISPR tool in relation to neurology. The paradigm would also involve variables that need further investigation. Furthermore, it would eliminate biases and focus on truths of information retrieved while collecting data and evidence.
The post-positivist paradigm is used to gather data and evidence based on observations and trials that has been proposed regarding genetic therapies on the treatment of neurological diseases. This paradigm constitutes a single truth through the process of trials in determining the benefits and risks of the CRISPR-Cas9 system. One of the scientific processes conducted to prove this point is through various tests wherein scientists remove some human cell in order to in order for one to be able to edit the DNA sequence. This is then injected back into the body to test if its defense mechanism to certain diseases depending on the disorder being targeted and how it is able to combat the causes leading to the disease. In many cases, it is essential for researchers to conduct and analyze information based on theories and not only directed on a single person. It is likely practical for them to look into this thoroughly when dealing with the safety and efficacy of a particular study and its applicability to patients. In medicine, it is important to determine and develop general laws. The concluded truth of these trials are to be derived from the results and not inquired from personal beliefs and data.
With the traditional view of the post-positivist paradigm through research and trials, the results from the data collected will allow the researchers to understand the effects and potential of CRISPR gene editing in relation to neurology. This paradigm best applies to the phenomenon selected, as the researchers can come to a definite conclusion on the benefits and risks of the CRISPR tool, and will be able to measure the impact of each benefit and risk neurologically. Since the paradigm looks at the case with the idea of it having a definite truth, the researchers will be looking mainly into whether the benefits outweigh the risks of the CRISPR-Cas9 in relation to neurology.
The other three paradigms are not as appropriate to use for this research topic, considering that they take into account the background or beliefs of the participants and require the researcher to have a theoretical point of view in analyzing the situation. Therefore, using other paradigms would demand the researcher to think of a problem subjectively, which is not the goal, since the chosen facet is focused on determining the causes that influence the truth about the effects of CRISPR gene editing on the brain through careful observations and evidence.
Using a post-positivist paradigm with a qualitative approach would require analyzing non-numerical data such as evidence acquired from DNA gene editing testing. The research would be based on effects from the testings to gain knowledge about the benefits and risks of gene editing neurologically.
The main objective of the research problem is to conclude the effectivity and efficiency of gene editing when it comes to therapies on brain disorders. These series of trials are done through qualitative case studies, through descriptive explanations grounded on theoretical framework, an example of which can be seen in proving and analyzing the use of CRISPR-Cas9 tool to identify antibodies targeting Cas9 proteins. The aim of the research possess direct description on qualitative data rather than statistical views. Majority of the studies regarding this particular topic have utilized qualitative approaches in order to facilitate the discovery of the claims.
By approaching this research problem through a qualitative approach, instead of looking at numerical measurements or statistics, the researchers will look at each description of the positive and negative effects of gene editing on the brain. This approach will allow us to interpret the data in the most accurate way possible, by looking at the context of each data rather than making conclusions from statistics. Evidence to make these conclusions will be collected through DNA gene editing testing.
The qualitative approach, the quantitative approach rather, is not suitable to use for this research problem since the researchers aim to acquire information that is relevant or applicable given the research topic at hand, which is to identify the advantages and disadvantages of using the CRISPR-Cas9 system on the brain. Statistical and analytical data would be irrelevant for the research. Consequently, qualitative data would be best to use for the given topic.
Thank you for listening.